A very good afternoon. Welcome to the second session. In the current session, Dr. Prashant Karunakar will be delivering a talk on chemoinformatics with specific reference and emphasis on molecular docking. Dr. Prashant Karunakar obtained his master's in bioinformatics from Kuvempu University and PhD degree from PS Institute of Technology affiliated to Kuvempu. His research topic was crystal structure and molecular docking analysis of furan derivatives having pharmaceutical significance and determination of assembly of chains in protein targets. Prior to joining PSIT, he worked as a research associate at Indian Life Technologies Bangalore, where he performed target identification, validation, and docking studies of different ligands for their pharmaceutical significance. He also worked in two famous international synchrotron facilities like EMBL, Germany, and ESRF, France, where he focused on automating the number of monomers in the asymmetric unit of protein crystals and analysis of X-ray data and resulting structure from UV-induced radiation damage phasing. He has more than 30 national and international publications with good impact factor in Scopus Index journals. Dr. Prashant was the only candidate to get selected from India for the prestigious EMBO practical course on computational structural biology in April 2014 at the European Bioinformatic Institute, Kingston, UK, with complete travel support. He has completed a course on symmetry in the solid state conducted by Professor T.N. Guru Rao at Solid State Structural Chemistry Unit, IISc Bangalore. Currently, his research work is focused on identification of targets and lead-like molecules for Febrile disease. He is also conducting active research on FFAR2 and FFAR3 in collaboration with researchers abroad. Over to Dr. Prashant for his talk on chemoinformatics. Thank you.
Thank you, ma'am. Thank you for the introduction. Yeah, as Kumar got some emergency, we got to just prepone this particular talk, which was scheduled tomorrow afternoon, but it's fine. So before we go for working with the chemoinformatics, so I just want to know, like, how many of you are familiar with chemoinformatics or related to like bioinformatics aspects. So if by any chance, is there anyone who used with the bioinformatics applications or docking? They can just tell me something about what did they do. Or I will assume that you're not familiar with any of these things and will start doing from the as a beginner, and then we will move on to tell how we can identify a lead molecule. So in this particular session for 1.5 hours, whatever we have, we will work out with how exactly the drug discovery is going to work. That's with the introduction of drug discovery. Then we will move to the science involved in disease target identification, then how we can perform the virtual screening, then how we can analyze this virtual screening result, and then how we can decide this would be the possible lead-like molecule. Then we will also, whatever the results we will get with the virtual screening, right? That goes more or less with molecular docking, and how we can analyze the results and how we can analyze the hydrogen bonds, how we should analyze the LigPlus. So more or less these things will be covered in the entire talk. So as I mentioned, we will start as if we do not know about this, because looking into the participants list, we have all variety of participants. Few are from the pharmacy background, few with the botany background, few are with the chemistry and biotechnology. So you may be heard of but not used the tools, or maybe willing to use but don't know how exactly the work you have done. You can describe your work with your collaborator and further use it for your research. So what I thought is I will give the overall picture of what exactly this drug designing is, and then you can just think how best your work you can extrapolate for the docking or dynamics or other aspects. First thing, you will also get a doubt like how is this drug discovery or chemoinformatics uh, in the NGS workshop, right? So as Kumar also described in the morning, and when he mentioned about the amino acid change and the protein responsible for the diseases, right? So if when you do the sequence, let's say the genome sequencing of the patient, and then if you identify the kind of a mutation that is there in the patient sample, so it gives a clear hint that there is some amino acid change that has happened. Even in the morning session, he showed the amino acid change table. So that amino acid change provides a hint that there is a change in the protein structure. So when there is a change in the protein structure, so we got to understand like protein are the functional units. So they are the one who works for our body to do its normal activities. So if there is a change or uh, cannot function, right? So there is a need from the external agency to repair that or activate that protein to do its normal activity. So in few cases, the protein is not supposed to work. That is something like overexpression, but the protein is functioning. So again, from the external, we got to put some molecules and make the protein not to function so that we will control the disease. So in that way, if we look into, we have two types of work. Either we got to make the protein to function if it is not functioning, or if it is overexpression or working a lot than what is required. Again, the lead molecule antagonist has to bind and then control its activity. So in the overall study in the chemoinformatics or disease or any work, if we look into, so the important aspect is with the drug. So drug molecule or the this drug plays an extremely important role in controlling the disease. Now to get this drug molecule, right? So there are two ways by which we can get this. First method is like structure based drug designing, and another method is referred as ligand based drug designing. Now in the structure based drug designing, we will be aware of the protein structure, but we will not be knowing which ligand going to act like a drug molecule. Then we will use a particular, this is the method structure based drug designing, and then we will get the lead molecule. So just to give a kind of an analogy between this SPDD or structure based drug designing and ligand based drug designing. So the easiest way is, uh, let us say you want to open a particular door of any example you can take. Okay, even in our department itself, or let's say my cabin. So I locked, but I forgot to bring the key or I lost the key. Just an analogy. But I want the key to open that. But the lock is already there, key is not there. So that means the door, which door I want to open, that aspect is very clear for me. But the key I am not having, so I lost the key, for example. Now the lock is nothing but the protein. The key is nothing but the ligand. Now what we do is we will bring the key maker, and the key maker will look into the lock. Then he will say like, okay, this is from this company. This is having a 5 lever or 6 lever, and these type of the keys are going to work. Then he will shortlist the number of keys, and then he is going to open with some trial and error, or sometimes he may need to modify a bit of the key, and then he will open the door. Now this process, if you can just imagine the entire process. So the lock is already there, and that means the protein is already there. The lock, whatever he brings in, it may open 50%. That's nothing but lead identification. He will make the optimization. That means the key opened the door 50%, but he has to make some modification in the key so that it will open completely. So that process, whatever he does in trimming of that key, that's nothing but lead optimization. So once we optimize, then it will open the lock completely. So if we just look into that process, right? We have the protein structure from the PDB, then we will cross check, that is whether the key, whether the lock is correct or not, we will validate. Then where should we put that key? That's the binding site. Then we will put the key. Now he may come up with 1000 keys. Just like that, we may get millions of compounds from zinc database or drug bank or keg or pubchem. So we will use all the ligands, whatever is there. Before we use all the ligands, we can put which are the keys which are going to cause toxic effect on the body. So estimation of ADMET will be made, then reduce the number of compounds, then do the docking and further analysis will be made. Now this is a kind of a okay, like you know the door to open, but the ligand based drug designing is a slightly different aspect. It's something like I would say the pharmaceutical company usually works with the structure-based drug designing, and the academic sections or academicians majorly work with the ligand-based drug designing. So what exactly this ligand-based drug designing? If you can look into the profile of most of the academic researchers or anything, so they are working with one kind of a series of ligands. So you will keep even in my PhD. So we work with furan derivative. So only the furan derivatives we will be keep on producing, and then check what are the other biological activity this furan derivative is having. That means we are more or less targeted to the ligand, and then check what are the other biological activities this ligand can perform. So the center of our research will be towards the ligand. But in the structure based drug designing, I don't mind which key is going to open. Finally, I want that door to open, right? So that is why that means the protein centric. So that is why it is structure based. So protein structure is the center. Here the ligand is the center. We are more or less working with the ligand. Now, how is this going to work? So we have a key, let's say, and take that particular key and check all the doors that are there in your floor. That means take the key, make that key to dock with all the existing proteins, ligand with all the proteins, then check to which particular protein this ligand is binding. In other words, to again coming back to that analogy, check which door this key is going to open. If by chance, if that key is going to open any room which is of some significance, then your key got more value. So you will claim like we have designed, at the end the claim will be like a new molecule is designed to inhibit anti-diabetes, developed an antibiotic drug or anti-tubercular drug or anti-cancerous drug or antibacterial, antiviral, like that. So did you did you work on developing anti-diabetic drug in the beginning? No. So you just designed a molecule, and by chance that molecule is having anti-diabetic activity. So, but what about here? We started to develop a anti-diabetic drug, and we screened all the molecules and come up with a new ligand which is going to control the diabetes. So that's the very thin layer difference. That means your target is specific here, but here your key is specific. So once we have the small molecule, we can check its properties. We can check with its all possible descriptors, then check how is the relationship with other ligands. Let's say I have one molecule and how many such similar ligands are there. It's something like I have one key and then take out all the keys that are there in the department, for example, in our department. So to which particular key, the key which I am having is matching with. So if it is matching 90% with another key, so that gives a clear hint that, okay, so my key is matching 90% with the existing key, and this is opening this particular uh, door. So it provides a hint like what would be the uh, another door or the same door that it can open. So that is where this support vector machine or multiple linear regression models, all these things provides a hint. One can also conduct this QSAR study, that is the quantitative structure activity relationship. So you have the structure, you want to know its activity based on the quantity, then bring up the relationship. You have the ligand structure based on the existing activity information of other ligands, how much it may match, then bring out the relationship. That's the QSAR that's going. Or once your structure based drug designing is done, you have made the binding site, look into the pharmacophore information, then also you can start performing the docking based on the ligand information. So after evaluating the correlation, weighted models, and support vector mission, all these information, you will get to know like, okay, your lead molecule or your ligand that you have selected is going to have this particular biological activity or bioactivity prediction. This is majorly the docking. In either of the cases, that's where the docking is. Once the molecular docking is done, then the next step is it goes to molecular dynamic simulation. So what is the difference between this molecular docking and molecular dynamics? So molecular docking is a kind of a, just to again bring the easy understanding, molecular docking is kind of a taking a photo, and molecular dynamics is a kind of a capturing a video. So there are some advantages with the docking and there are some advantages with the video, right? So we'll get some additional information in video compared to the photo. So in the same way, once we have the dynamic simulation done for that protein ligand complex, uh, usually like 100 nanoseconds simulation time, then we can perform the RMSD, root mean square deviation, root mean square fluctuation, radius of gyration, DSSP analysis, energy calculation, MMPBSA, displacement analysis, torsional angle calculation, principal component analysis. So many other analysis can be made. Then finally, if we have like 5 lead molecules, for example, after docking, so all 5 can be considered for dynamics. And then finally, you can say like this would be the potential lead molecule. So you will not make a conclusion after doing this dry lab work, like potential drug compound. So our conclusion will be saying like potential lead like molecule or potential lead compound. So if we perform only the docking, you will say like potential lead like molecule. If you perform dynamic simulation, you will have some more confidence, then you will say like potential lead molecule or lead compound. If we do the in vivo in vitro analysis, you will have more confidence. So you will say like probably drug like molecule. After the animal, after the human clinical trials and all the other stuffs, and if the FDA approves, then the term drug will be used. So till that particular time, it will be like lead compounds. Now this is the entire picture of chemoinformatics as a whole. So if we are particular with the ligand and want to check its activities, then go with the ligand-based drug designing. If you are particular about the disease or the protein, then you go with the structure-based drug designing. So based on what you are working with or the what we have as a input, one can take a decision whether with SPDD or LBDD. But to perform this simulation, we need this complex. And to get this complex, whether you follow with this or this, either of the ways, finally we need the complex to do this MD simulation. So this is a general slide which more or less all will be familiar with. So one has to identify a disease, first one. That's typically even in the pharma industry, that's how it goes. Identify a disease, then identify the protein which is involved in that disease, then isolate that protein, then do the cloning, purification, expression, and then crystallization, get the structure of this protein. Then once we have the structure, find a drug effective against this disease protein. And whatever we are working out now with reference to molecular docking, that is where this is where the entire uh, process comes into. If the crystallization is going to take time, so through bioinformatics, we can predict the structure using homology modeling. So once we get the homology modeling done, model built, so take out the ligands from different databases, as mentioned here, PubChem, DrugBank, Zinc. Then we'll have like sometimes millions of compounds. Test each of these molecules because it is cheaper in the computer. So get the simulation done, then identify the lead molecule. Then further it goes to preclinical, then formulation and scale up, investigational new drug, then human clinical trials, then the new drug application. And if the FDA approvals, then it will be called as a drug molecule. This is the entire process. So our role in the entire drug discovery is here. Sometimes after the preclinical testing, if the ligand, the lead molecule brings some side effect, again that ligand molecule will come back to this, do some modification. That's where the I mentioned like the lock is opening 50 or 60%, then the optimization needs to be done. So again, so this is where the intuition or the entire skill, what because the skill of the key maker comes over there, like 50% if it is opening, where he should trim that key so that it will open the lock. So to do that, he should understand thoroughly about the lock and also about the key. Both should be very, uh, he should be experienced with that. Then he can make the modification. So like what functional group we can place, change it in this ligand so that it can clear this stage and go into the next stages and finally become like a drug molecule. So typically, it takes like 10 to 12 years time to have the drug molecule into the market. And in the beginning, it will be like having some millions of compounds, and then active compounds will be like 2000. Then we'll have true hits like 200, lead compounds, that is at the animal. Then the clinical trials 10. And if the pharmaceutical company is lucky, right? And then we may have one molecule coming out to the market. So 10 to 100 researchers will be working on the drug discovery project, and 2 to 10 years it will take. And some projects never even get started because there are not adequate assays or animal models to test. So this animal testing or the assays, all those things should be known before starting the project. Is there any adequate assays available to test? Because you will, let's say in this case, you will design a molecule, but what if there is no animal model available to test? Then we cannot go to the next stage. So those things has to be taken care. So in entire study, the two terms are usually used, that is the efficacy and then activity. So efficacy, that is nothing but like effect. So it's a qualitative term. So the qualitative property of a compound having the desired effect on a biological system. So whether it has effect. What is the quality? Activity is a quantitative measure. So quantitative measure of how much of that compound is required to have the measured effect on the biological system. It's something like will be like whether, let's say you take um, some powder, for example, and then if you taste, then if you say like yes, it's sweet in taste, right? So you got the qualitative property done. Then you will also ask like how much I should pour, right? How much I should pour into, let's say if you are preparing a tea or a coffee or anything, how much I should pay, I should pour so that to get that desired or required effect into that one. That's where the quantity will come into. So 1 cube or 2 cube of the sugar, if you put, that brings this much of the sweetness. So where sweetness is there, that's the qualitative property. How much of that sugar cube you should put, that's the quantitative property. Both are required. So target is usually a protein, but in some cases one can do DNA, RNA, and another biological molecules like carbohydrate and other molecules becomes like a target. But in the most of the drugs that are there in the market, they more or less around 80% of the drugs will bind to the proteins. And drug activity is obtained through inhibition of the target. That is also a most of the cases. The inhibition of the target or the protein will be done in most of the drugs activity. So looking into the terminologies, when what term we should use, right? As I mentioned earlier, hit refers to compounds identified in some initial rounds of screening. So as here, that's something like hits. So even all these three together is like a hits. So the number of molecules that you consider in the beginning. So that's the thing, but the hit compounds identified in some initial rounds of screenings. Again, it will be in millions usually. Then you will do some screening and then you will get into lead molecules or leads. So that is a series of compounds meets a certain set of criteria. So you will put some set of criteria. Again, I will also mention the criteria in the later point of time. Like this should bind to this protein, or this should have the binding energy of this much, or should have two hydrogen bonds at least with these two amino acids of the protein. So those are your certain set of criteria that you will place. If it follows, if it meets all those criteria, then you will call those molecules as the leads. Then these lead molecules will go into the preclinical, clinical, and further goes into the approval for the drug-like molecule. So the approval has to be given in India like CDSCO or the FDA in USA for treating an illness. Then the term drug will be used for that. So how is the typical um, procedure, right? There are different ways by which one can design your experiments for the drug development. But one of the most widely used method for the drug discovery is the competitive inhibition. So because it provides some hint about how the key should be. So that's one of the advantage. Let's say there is a reaction happening, the enzyme is there. So enzyme is nothing but a protein. So that is why most of the targets are the proteins. So enzyme is already there. We have the active site of the protein, the substrate binding to this, and then once it binds, there are some structural change in the ligand, but the product formation is happening. Now let's assume a situation. Uh, we have an enzyme and we have the substrate binding to this protein, but the product formation should not happen. Okay, in other words, if it is happening more than the required, that's where the disease is going to occur, right? Anything which is more than the necessary. So if the protein expression is happening more than required, that's going to cause the disease. Now we need to have an inhibitor. This inhibitor is nothing but the drug molecule. So this should bind to the enzyme and block this active site, and the substrate should not bind to this active site. So when the substrate is not able to bind to this active site, then the product formation is not going to happen. In other words, the disease is under control. So what is usually done is most of the time it is the mimicking property will be used. So use the substrate structure and look for the analogs. If you just look into this, so this portion and this portion is similar, looks same, only the difference is over here. Now, when this reaches to the active site of the protein or the enzyme, so enzyme feels like yes, the substrate has come, so it will just take that one. So it will accept, and this binds to the protein. Once it binds to the protein, because it is not the substrate, so the product formation is not going to happen. Although the substrate comes over here, but since the inhibitor is already bound to the protein, so the active site is blocked. So substrate cannot bind. So the uh, product molecule is not going to get generated. So this is the competitive. So that means this inhibitor is competing against the substrate. Before the substrate can bind, the inhibitor is binding to the active site protein. Now, if you imagine this situation, right? So one has to understand about the substrate and the enzyme and how much energy it requires to bind. Now, when I mentioned like our inhibitor should compete against the enzyme and bind to this protein before the substrate can bind, right? So these are all seems to be like relative term. How will we get to know like when substrate will bind or how best it will bind? What is that parameter I should use to get to know like our ligand molecule or inhibitor is better than the substrate, right? So to understand all these things, uh, the binding energy or the Gibbs free energy is used as one of the parameter uh, to evaluate whether the inhibitor binds much better to this enzyme than the substrate. So lower the energy, better is the binding. So then you are making the competitive inhibitor to the drug. So a drug should bind to the active site. The positioning of the functional groups in the drug molecule will be made, and then we have the create drugs that are transition state analogs. So that's how the competitive inhibitor is going to be designed. Now let's look into another aspect, that is the target identification. So if you recall this particular slide, right? So this is first is disease identification. This target identification is nothing but selection of the enzyme. So whatever the enzyme we are mentioning here, that's nothing but the target molecule. We are targeting that molecule. That's why the term used is target identification and characterization. Now, how can we target that particular protein, or how should we select the target first and then use it for the study? So there are different ways by which the target can be identified. I will just show you some of the examples how one can do. So to do that, one should understand how exactly the disease is spreading or how is the disease is occurring in the body. So if we take some example, let's say like HIV virus, which is going to cause this HIV AIDS disease. So one should understand how exactly the structure of this virus is, and then understand where it binds and how it enters into the human body. So if we look into this, so HIV virus, the GP120 of that protein, GP120 protein binds to the CD4 receptor of the human cell. Then it binds to the CCR5 protein, and then it enters inside the body. It's something like this. So if this is the cell and if this is the virus, first it binds to this, that is the receptor, then binds to this and then enters inside the body. So CD4 is one receptor, CCR5 is another receptor or co-receptor. So by understanding this uh, disease, how exactly the virus enters into body. Now we have two target proteins, CD4 or CCR5. Then one can look into again where it is present, whether the between these two, which one is better to consider, what would be the probable side effects if I inhibit CD4, what would be the probable side effects if we inhibit CCR5, prioritize between CD4 and CCR5, then one can take a decision and then work upon this. Now, once we fix with this, let's say CCR5 as our target protein. Now, this becomes like our uh, structure, which can be, so this becomes like our structure based drug designing, because we are fixed with the structure. Protein we are fixed with. Now we are looking for identifying the ligands. So that is the structure based drug designing aspect. Like this. So we have this CD4, then the HIV, then this is where the GP120 binds and enters into our body. Then we know, like, yes, we'll use this, develop a antagonist, co-receptor antagonist, which will bind to this. So this is the active site. For example, if this is the active site, so the ligand or the inhibitor is bound to this so that the GP120 cannot bind with the CCR5. When it cannot bind, it cannot enter into the body. So that's how the target identification is done. Even if we are working with the pain and inflammation, right, one can look for cyclooxygenase 1 and cyclooxygenase 2 enzymes, then look for how exactly this COX-1 is, COX-2 is, and how is the arachidonic acid is going to bring the effect, then understand its pathway, then take a decision whether to work with the COX-1 or to work with the COX-2, then look at the protein and then develop the drug molecule. Even for the antitubercular, if we are working there also, it goes the same thing. So in the major global infectious diseases, that is with the antitubercular, so one has to understand this slide is going to provide information like we should know about the disease, like what is the rate of deaths that is happening because of that particular disease. So in case of pharmaceutical industry, this slide is a very important slide because they want to see if this is the number of deaths, or this is like 8.7 million people fell ill from the TB in 2011. That means these 8.7 patients are their customers. In other words, that means 8.7 million tablets or whatever the drug will be selling product for them. So this is an important aspect with reference to pharma, and this also provides information like at the geographic level, where are their customers are, in the patients, where are they are present, how much it is. So in that context, this slide provides information like um, geographic information and how many will get infected with that, what is the age group, everything needs to be analyzed before selecting a disease. Once we decide the disease, then we have to understand about the signaling or the pathway analysis. Okay, how exactly this tuberculosis disease is spreading, or how exactly it is getting infected. So where exactly it is happening, what is the role of the sensor histidine, what is this two component signal system, or how is the there is like one sensor component, another one is the response regulator. So the signal will be sent, then the uh, autophosphorylation is going to happen, then it will send the signal. This aspartic acid is going to receive the signal, then response regulator is going to have response regulator or the output further to the DNA bind. So this scheme provides information like how is the signal transduction is happening. Then one can go in detail at the next level, at the pathway level. Here it is just like an overview. Now, where exactly this signal transduction is going to happen, and then which protein is there here as a receiver, right? So at the protein level, this is not at the protein level, this is just an overview. Once we go for KEG pathway, there are many. This is the Q2 Encyclopedia of Genes and Genomes KEGG pathway. This will provide a clear hint like, okay, so NARL protein is the receiver, which receives the signal, and then the further signal transduction is happening. So within this NARL, how it works, right? So what we, if you can just imagine yourself, so we are going down to a level like at the geography about the disease we will understand. Then we will select the disease to be worked out. Then from that disease, how exactly the disease is getting spreaded. Just if we imagine this one, same like this, whatever we have put, the virus is there, then the CCR5 protein or CD4 is the protein. Then it enters inside the body. In the same level, this is how the signaling is happening. Then if we look at the protein level, then we will get the protein information. And then these are the proteins, whatever is there in the green are getting expressed in that particular organism in the Mycobacterium tuberculosis. Then further we looking into this NARL, right? We will get the key residue. So the phosphoryl residue is then transferred by the kinase to an aspartic acid, aspartate residue ASP61 in the receiver domain of NARL. So that ASP61 is what this one is, ASP. So we are more or less to the protein to a specific residue at the amino acid level we will derive. Then once you take out this NARL protein, download from PDB or anything, then if we look into the protein, right, like 3EUL is the PDB ID. Then if we look into the entire protein, where is this aspartic acid, ASP61 is, which brings about this uh, autophosphorylation happening, right? That gives a hint like, okay, this is where the site of interest is, ASP61. That means you will get a clear hint like, okay, I have to design a drug molecule that will block this site so that the autophosphorylation is not going to happen. In other words, you are going to bring about the uh, inhibitor designing. So once we have this, right, so you know the residues that are surrounding to this protein, how big is this particular active site, right, and how should be the ligand is. Then it helps us in designing the lead molecule. So one should also look at where exactly the protein of our interest is present in the body. So if the protein of our interest is there in the central nervous system, right, then we got to design our lead molecules to cross this blood-brain barrier. So that is why usually the target selected is like extracellular space. So if the target is there in the extracellular space, even we don't need to think of making the ligand to cross this lipid bilayer. So if it has to cross multiple layers, so the complexity increases. So designing a lead molecule becomes too difficult. So usually the proteins, that too the extracellular proteins, if it is available, it will be considered as the targets. Now, if you can imagine whatever I was mentioning, right? So this is the site. If we just imagine like this, so if the ASP61 is here, and if we want to block this one, so one can easily calculate what is the distance from here to here, from here to here, here to here, here. So then you will get a hint like how big should be the molecule. It's something like, again, if I bring back, if you look into the log properly, then the key maker, if you can imagine, if we by chance, if you have come across this particular situation, if in your house the door is not opening or lost the key, so the key maker may bring 1000 keys. It is not like he is going to test all 1000 keys. The moment he looks into the lock, he will decide like these 500 keys is not going to work, right? So he will just keep those 500 ligands apart, and then he will keep the 500 rest 500. Then he will look into the lock again precisely. then he will again remove 100 or 200 keys. Then he will get into 200, 250 keys. If he thoroughly looks into the log, so the number of keys getting rejected or keeping aside increases, then he will take, that is what his experiences. Then he can use some keys, then he will make the trimming and then get the drug molecule. So that's where exactly the pharmacophore is also becomes an important aspect. So if your active site is something like this, just an imagination, this is similar to whatever I have put here. So you know the residues. So if you know like this is the amino acid, this is another amino acid, this is another amino acid, three amino acids which are there, and you want your ligand to interact with all these three amino acids. Then you know like okay, what is the property of this particular amino acid? Okay, polar amino acid, non-polar amino acid, acidic amino acid. Then you know like okay, if this is the property of this amino acid, what functional group I should keep here so that it will bring the complementarity between them, so that this functional group will interact with this amino acid. This functional group of the ligand will interact with this amino acid, and this functional group will interact with this amino acid. Now the term I use is something like interaction. That means the interaction can be by making the hydrogen bond or hydrophobic interaction, or there are many such interactions that are there. Formation of the water bridges, pi-pi bonds, but there are different levels, van der Waals interaction. So whether this functional group is having a complementarity with this amino acid. Now, the amino acid cannot be changed. What is there in our hand is we can change this functional group. So keep changing the functional group, right? So that it brings the complementarity to this amino acid. Once you know like, okay, these three are the important aspects, then use the linkers, link these three functional groups and design a drug molecule so that it will control the activity. Something like you can have this molecule as a drug molecule that which can interact with these three amino acids, 1, 2, and 3, then cover this active site so that the autophosphorylation is not going to happen. In other words, you inhibited this particular protein. By inhibiting this particular protein, you have further controlled the disease. So that is how the entire work goes. Now, just to uh, bring about, so this is the theoretical aspect about disease identification or target identification or pharmacophore or lead molecule identification or whatever. Now let's assume like we have a protein, right? In other words, again to bring it to easy understanding, I got the key and I bought some, I bought the log and I have come up with some keys, but I want to test whether the key is going to work or not, right? So since in the door and the lock and the key model, whatever we discussed, so the key is going to placed and then tested. That can be done in uh, in vivo and in vitro also. But how many such ligands you can test, right? So it becomes a very expensive experiment if you want to do with the biochemical assay or cell-based assay. So it's going to take years of time if we want to test, let's say, 10,000 molecules, and the cost, labor, right? So it becomes very expensive. That's where the alternative method has come into picture. That's the protein ligand docking. So what is done here is, you know the log, right? Can I make the 3D of this log? Yes. Then you with the entire log, you know where exactly I should put the key. That's the key region, right? So you build the log and then you know where is the place for I should put the key. Then that becomes our active site. Then do I really need to test all the keys to this particular log, or is there any other way by which I can test? right, virtually. So then, yes, build the 3D model of this protein, okay, rather than testing in the cell-based assay. That's where the crystallography or NMR comes into picture, rather than just doing in a realistic manner, something like testing directly into the NMRs, right? Or clinical trials or anything, right? Or cell-based or biochemical assays. So let's get the 3D model of the protein first. So crystallographers will develop the 3D model of the protein and they will determine the structures, and then you will get the 3D tertiary structure of the protein done. Then once we know the tertiary structure of the protein, then we can also get to know, predict, or if they do the co-crystallization and then determine the structure, you will also get the binding site. Now we know, like, okay, this is where exactly the ligand should bind. So that information we will get. Then once 3D is a, again we can look the 3D structure of the protein in a computer, right? So and also we have the 3D structure of the molecules, ligands. Then we will use some tools to wherein we can place the protein as it is and keep the ligands going inside or interact with the protein, come out. Take the second ligand, go inside, interact with this protein, come out. So test like this. So whether we have 100 ligands or 1000 ligands or 1 million compounds, it is not a, because in silico or doing it in computer is very fast and inexpensive process. But yes, there will be challenges like accuracy, because when you test with the biochemical or cell and assay, your accuracy will be very high. But here there will be a less accurate, but you don't need to do. It is not as poor as like 10-20% accuracy. So 60 to 70%, 75%, or I will also demonstrate in my case how is the accuracy is with the prediction. So we will get a rough estimate. Then you will say like, don't use these ligands. So in the entire process of the drug designing, okay, protein ligand docking, we will not work out in selection. We will work out in rejection. It's something like, how many molecules do you want at the end of the drug design, right? Or the drug discovery molecule. If you look into this particular chart, right, we want only one ligand. We will get only one molecule as a drug molecule. But how many you started with? We started with, let's say, 2 lakh molecules. Now, what did you do from 2 lakh molecules to one? We did not work out in selecting one. We worked out in rejecting the compounds. So your selection criteria or rejection criteria has to be very strong. You will test and you will reject. So what did we do from 2 lakh to 2000, right? So 1,98,000 compounds we removed with some criteria. From 2000 to 200, we have put some criteria to reject. The compounds which fulfill this criteria is nothing but the selected, but our criteria is more or less towards the rejection. So in other words, if I have to put this into an analogy of selection, we may have like 2000 candidates attending an interview, but you want to hire only one candidate for that particular position. You will not work out the criteria for the selection. You will put the criteria to reject the candidates. Something like his percentage in 10th and PUC should be more than 80%. He should be good in group discussions, technical skills, all those things you will place. And those who are not good in doing that, you will keep rejecting them. Then one student may proceed with all these steps and get rejected. So in the same process, right, you may have 2 million compounds, but you will keep rejecting them and select the one which fulfills all the criteria. Once it fulfills all the criteria, that becomes like a good drug molecule. So in the protein ligand docking, it's a molecular modeling technique which predicts the position and orientation of the ligand. We are going to check where it binds to the protein, how it binds to the protein. Both are important. That's something like Given two molecules with 3D conformation in atomic details, that is the 3D, or up to the atomic details, do the molecules bind to each other? If yes, let's say protein and ligand. Those two are the molecules. If yes, how strong is the binding affinity? That is the quantitative. How strong is the binding affinity? -6 kilocalories, -8 kilocalories, that one. And how does the molecule complex looks like? How exactly the complex looks like, right? Visualization. That's another important aspect. So what are the problems that we can address through molecular docking? Majorly done is with the protein ligand docking. Widely used is this. Within this, one can keep the flexibility for the protein and or keep the no flexibility for the docking or the protein. That's the rigid body docking. Or perform protein docking, protein DNA docking, DNA ligand docking, ligand carbohydrate docking. So in my case, I worked out with all these cases with the different collaborators. Protein ligand is done, protein is done, even with the proteins having like 600, 650 as 800 amino acids. Protein DNA docking is also possible to perform. How the ligand binds to the DNA. And when it comes to DNA docking, we got to test like two different ways, whether the ligand binds to the minor groove or whether it binds to the uh, minor groove or major groove and intercalation, right? Is there any intercalator possibility? So it's slightly difficult to perform, but yes, and also with the dyes, how exactly it will bind to the carbohydrates, right? Or matrix creation with the cellulose matrix creation and how the different dyes binds to these fabrics and then how we can remove the coloring. So carbohydrate ligand docking is also another kind of a study that can be done. So this is the just a representation one can do whenever it comes to like molecular docking. We are working more or less towards the structure based drug designing. It helps in lead optimization, high throughput screening, and chemical mechanism studies, how exactly the ligands are functioning. So whenever the ligand binds to a protein, so these two are the molecules, biological molecules, protein if we take and the ligand, and the complex formation is going to happen. Now, one of the important term that is used is this delta G. So this delta G gives information like how best is the complementarity between these two molecules. So this increases the delta G. In other words, if usually it is in negative term, -7. So one has to remember that minus anything. For example, -6 is better than -5. Okay, it's a relative term. There is no threshold saying like anything with -7 is always good. No, it is just a relative. So it's a relative term. If the existing drug is binding at -2 kilocalories per mole, and if your ligand binds with -3, then compared to -2, -3 is better. So you can say like your ligand is better than that. So it's a relative term. So the delta G decreases -3 to -4, -5, -6 based on this complementarity. How best is this complementarity between these two molecules? So if the complementarity is not good, right, that's something like 50% opening of the lock. So if increases complementarity, right, it's a basically the lock and key should be complementary, then only it will open. So if it is opening 100%, that means you have developed a best ligand that can open the key, right? So we'll take thousands of ligands, take these receptor, do this, use different docking softwares, check where it binds, and then one can take a decision. So complex quality is evaluated with the score. That score is either delta G or bending score. If we are using glide score. So that's the measurement. Finally, the number. Okay, finally, one number should be used to describe it. So take the protein, take n number of ligands, whatever you want, then perform the docking. Then we will have the future drug developed with this procedure. Coming to the publications, how usually it is, right? assuming that there are many who have not done the docking or maybe thinking whether the docking is going to be helpful in their research for the publication or not. So in that context, I have put this particular slide. So yes, over a period of time, the number of publications with related to docking is also increasing. So this is a pretty old data, but this is the reference paper from which it is done. So by now it has crossed around 10,000 papers per year where they have used the docking software. So which tool to be used? Again, this is another usual question. Uh, there are many open source tools that are available free to use, but widely used is the Autodoc. So Autodoc is free to use and its accuracy is there in calculating that complementarity. And in fact, this Autodoc made um, all the academicians, most of the academicians to think about performing this in silico drug designing aspects and come up with the lead molecules. Then we have this gold, glide, FlexX, SurflexDoc, Fitted, Autodoc, Vina. Right, there are so many tools that are available for using for docking, but most are the commercial one. Autodoc is the free. Now to do docking, right? How is what exactly the flow chart that is there? So we need structure. First thing, if you can remember the first slide, right? Whether we want to do with the structure based drug designing. So we need the 3D structure. We can get from the drug PDB, protein data bank, then download that particular structure. If there are any heteroatoms or any other chains that are not required, so removal of water or structure protonation, all those things can be done. And if it is not there, use the homology modeling and perform the structure validation. Then if the structure is good, reasonably good, take it for the docking. Now protein is ready. We want ligand as it's the same from this is also from the same paper. So use the different databases, get all the small molecules, or if you have synthesized a molecule, okay, in your lab, so you can draw that particular molecule and then we can convert that 2D into 3D and then apply different force field, perform the energy minimization and then use it for the molecular docking. So this is the sequence uh, that is like protein preparation and ligand preparation, then goes into a docking. Now, once we allow this protein and ligand to interact with the interact, right? So the ligand may interact if we take this as our protein of our interest. Okay, if this is the protein. So the ligand may interact over here, or ligand may interact in this particular position or may interact in this particular position. So all these three are the docking results. Okay, all these three are the docking results. But you will know like whether this is a correct or whether this is correct or this is correct. Then you can take a decision. We wanted the ligand to interact in this. And yes, you can go ahead and select this particular position. But these are the three possible positions where it can interact. Okay, so relative positions, this. Then it's referred as 3 degrees of freedom. Then once the ligand is interacting within this active site, let's assume like this the active site. Within this active site, it can have three different orientations. This is one position that's at the active site. Here it can interact like this, or interact like this, or interact like this. So there are three different orientations. So how should we decide like whether this is the correct orientation or this is the correct orientation or this is the correct orientation? That's where this delta G or the Gibbs free energy or Vina score or glide score, whatever the tool you are using, it will be generating a value. So this will be having a better energy or score compared to this, and this will be better compared to this. Then you will know like yes, so this is a better orientation of interaction compared to these two. Then one can also perform like giving a flexibility for the ligand, because the ligands are also having flexible in nature, rotatable bonds will be there. So then it will add the complexity for the docking. That's like n degrees of freedom. We can also provide flexibility for the protein also. that is like m degrees of protein or degrees of freedom for the protein. So the amino acids and its bonds can also make a movement, side chain movements. So if we look into right, the complexity is increasing. So we are working with the position. If we find the orientation, yes, it is interacting at the active site. So that's a good hint that it is providing. But along with the interaction at the active site, we also need to check. I think someone is required to mute. Not finding them. Okay. So if we look into right, so with the first, if we identify the position, this is not sufficient. We got into position to orientation to flexibility of the ligand, then flexibility of the protein. So the ligand should work out in a way, it should interact at the active site, right? And block should have a complementarity. So that is where the energy term plays an important role. So if this is the one interaction, then the energy is very high. But it is interacting at the active site, but not as good complementarity as this. So the energy is high. So this is the best complementarity, the energy is very low. So rather than checking all the conformations, in few cases, just looking into the energy, right, provides this E is the energy, N is the number of conformations. So this provides a clear indication like, okay, this is a better complementarity than the other two docking results. Now coming to one of the case studies, okay, this is a complete general aspects, whatever we discussed so far. So just to bring you into one of the case studies where I worked with in collaboration and to develop an agonist for this FFR2 protein. So I will just explain you how we worked out and identified a lead molecule as a overall, a kind of a case study you can just look into. So even in the NGS, when yesterday Dr. Debajyoti was mentioning, right? So taking these dietary fibers plays an important role in maintaining the good microbiota and then helps in the overall health of the person. So this study is also in the similar lines. We wanted to work out to identify the lead molecule, okay, which can control the disease and which can control the disease and then brings about the good health in the body. So normally this process is going to happen, but because of the lifestyle change and other aspects, the quantity of these small chain fatty acids available in the body gets reduced. And when this gets reduced, the activity of the key proteins like free fatty acid receptor 2 or 3, FFAR2 or FFAR3 gets limited when these proteins are not getting activated. So in other words, their job gets reduced. So we are looking for identifying an external agent. If it is with the drug, it is fine. Or is there any particular type of a food that if we consume, that is going to trigger these proteins and then brings about the balance in the entire health. So that was the overall concept. So dietary fibers, and then we will generate the gut microbiota, will generate the short chain fatty acids, and then it will further activate this FFAR2 or FFAR3 proteins. This activated FFAR2 or FFAR3 further helps in normal functioning of the neurophysiological function, adipocyte homeostasis, intestinal homeostasis, immune function, and pancreatic beta cell homeostasis. So this is what its function, overall function is. So same thing, dietary fibers with the short chain fatty acids, with the bone marrow, insulin production, or adipose tissues, peripheral blood cells. So even if we look into, so the expression of these FFAR2 protein, one can find it in the brain, bone marrow, heart, lungs, liver, pancreas, spleen, adipose tissues, intestine, muscle, right? So we can see the um, expression of this particular gene expression of this particular FFAR2 gene at different organs. So based on this, we got to know, like, yes, there is a need to activate this particular protein. Now, how should we activate this particular protein? Right, that's what the question is. And is it like we can with the food, if we can take some food which is going to activate this short chain fatty acids, that's going to be a much better way than synthetic method or having a molecules synthetic drugs. So in that context, again, it's the continuation of this, right? With the increase in the insulin resistance or lipid, it will activate by the short chain fatty acid receptors functionality at different body parts is explained over here. So overall, right, if we use these short chain fatty acids, the same thing like whatever we mentioned earlier. So there will be a decrease in the fat accumulation or increase in the energy expenditure in adipose tissues, decrease in the insulin sensitivity or increase in the glucose homeostasis in the pancreas. So these are different functionalities which is described over here. So based on all this, we got to know, like, we got to identify a molecule, and we also need to identify from which particular plant source it's going to trigger or activate this FFAR2 protein. So when we started working out with this FFAR2, right, there is no crystal structure available for this protein. So we got to work out. We took the protein sequences from the UniProt. We worked out with both human FFAR2 and mouse FFAR2 because this is the model organism. We cannot directly test with the human. So we want to see which ligand is going to activate work with the both, right? So we took both mouse and human FFAR2 with these corresponding sequences, performed homology modeling with the different servers available, ITSA, RaptorX, Swiss model, and many more. Then we look for the refinement of the structure, mod refiner or galaxy refine. So this galaxy refine is one of the tool available even in this galaxy server, whatever we were using in the morning session in the use galaxy. So we can provide the structure, and then further refinement of the structure is going to happen, like bond length, bond angle optimization, everything. Then we can check the Ramachandran plot with the ProCheck or Rampage, and then we can decide which model is appropriate. So before we go for any of the structure or structural analysis, it is extremely important that we understand at the sequence level. So the hierarchy usually goes like this: sequence to structure, structure to function. So that is why even in the NGS, that's at the sequence level, because whatever we say here, like arginine or any mutation that is there from leucine to tyrosine, whatever. So these are all goes back to a DNA level. So from DNA, then the transcription, then the translation, and then we will have the protein. So at the gene level, we got to understand that's what the NGS is helping us in understanding at the gene level. Then from that gene, what is its effect? That's what we are working out here. So yes, the translation happened and this coded for this particular amino acid. And then what is the difference between different proteins? Now in this FFAR family, right, how many proteins are there? We just said like FFAR2, I want to work with this FFAR2, but how many such proteins are there? So when we looked into, we have like FFAR1, FFAR2, FFAR3, and FFAR4. So there are 4 different FFARs that are there. Then first question comes like, is there any relationship between these 4 FFARs that are there? Then for first thing is perform the multiple sequence alignment, because these are multiple sequences. This sequence 1, sequence 2, sequence 3, multiple sequences will be considered, then perform the alignment, then analyze. So we got to know like, yes, there are conserved regions, right? Wherever the star is there, that star refers to conserved regions. This column refers to semi-conserved, dot refers to partially conserved. So it based on the amino acid properties. So hydrophilic, hydrophobic, that property will help in deciding. So yes, whatever the highlighted region is there with the yellow, that provides the information like how many helices are there. So one helix, second helix, third, fourth, fifth, sixth, and seventh. So there are 7 helices. So that's a 7 transmembrane GPCR protein. So that's the hint we are going to get. Now, if you look into right for a multiple sequence alignment, it becomes slightly difficult to make a conclusion about the relationship. So the phylogenetic tree helps us in deciding how exactly these proteins are correlated to each other. Then when we look into, so this FFAR2 protein is closely related with FFAR3 proteins. These two, that is what is this one. These two are closely related. Then between to these two, FFAR2 and FFAR3, FFAR1 is closely related. Then to this 3 FFAR4 is. So these two are closely related. Next to this is FFAR1 and then FFAR4. Now, when we started working out with the all the 4 FFARs, so very less information or some amount of information is available for FFAR2, very less for FFAR3, and still less for FFAR1 or 4. So based on that, we have decided, look, at least some information is available for FFAR2. So let's start working on this particular protein. That's how we started this project on FFAR2. Now we moved from FFAR2 to FFAR3. Now we are working on FFAR3 because it is not published. So we are still, I'm making the presentation on FFAR2. Now we also checked with the pairwise alignment. Since here multiple, that's why it is multiple sequence alignment. When you are taking 2, that is the pair, right? 2. So that's the pairwise sequence alignment. We checked how is the correlation between FFAR2 and FFAR3 in the human. Then we got like it's around 38% identity at the global end-to-end analysis, around 41% identity when we do the local alignment between these two proteins. So when should we do this kind of analysis is when you get to know like here FFAR2 and FFAR3 is correlated. Uh, what is the correlation or how much percentage of identity is there between these two? Because you will get some information for FFAR3, some information about FFAR2. If the FFAR2 and FFAR3 is, let's say, if it is 98% identity between these two, right? Then we can assume that both are one and the same. If 98, 99% identity is there between these two, whatever I will get for FFAR3, I can assume that it remains the same for FFAR2, or whatever I will get for FFAR2, I can assume that it remains the same for FFAR3. Now, just to get that, whatever we get for 2 or 3 is one and the same because both are 98, 99% identity. Can we assume like that and do our research? So to decide whether our assumption is correct or not, we had to do this pairwise sequence alignment. Now from this pairwise alignment, when you have like 38% or 40% identity, then it gives a clear indication that both are not similar. Okay? There is a huge difference at the sequence level between these two proteins. That means you got to work out or extract the information which is specific for FFAR2 as much as possible. That's the hint we will get. Then we wanted to work out with all the different species of this FFAR2 and FFAR3, because what we did here, right, it's only within the human, 1, 2, 3, 4 of the human. But we wanted to check because initially you will test with a mouse model. So whether human and mouse FFAR2 or human and mouse FFAR3, they are similar or how much is the difference between them. So perform the multiple sequence alignment and again generate the phylogenetic tree. So here we got to know like yes, the human FFAR2 is linked with the mouse mouse FFAR2. Here also if you take mouse and rat are closely related, but these two is correlated with the human FFAR2. If we want to do in the next stage with FFAR3, this also gives a hint like yes, mouse and human FFAR3 are closely related. These two are first level of correlation, mouse and rat. Next is with the human. So if we remove this, then there is a good correlation between them. Then it clearly provides a hint. Then the next question comes like, well, you got the correlation between human and mouse FFAR2. What is the percentage of identity between them? So we did the pairwise alignment between FFAR2 human and FFAR2 mouse. Then we got around 84% identity or 92.1% similarity. So that's also a good indication that there is a 84% gives a confidence that yes, the overall structure, whatever the mouse will have, will be almost similar with the human. So if we look for target for design a drug for the mouse may work with a human. So it's just the intuition that you will get because higher the sequence similarity, higher will be the structural similarity. That's because sequence to structure. Then we move to the structure. We build the model for human and mouse FFAR3. Then we superimposed both the structures. And once we superimposed, then we can get to know the RMSD between these two, how it is. So the RMSD, or the local RMSD and global RMSD, it gives like 4.16 angstrom is the difference when we take first amino acid to the last amino acid of both the proteins or few residues, that is from 40 to 143 few residues. If we compare, that's around 2.24. So anything near 2 angstrom, if we look into, they are considered to be highly correlated. So the two structures are highly correlated in their nature. That's the first thing we will get when we do the superposition. Then we wanted the ligand library, like our structure is ready. That's nothing but our key is ready. We want the keys now to test. So in our study, we took around 1,44,000 ligands for docking, and all this 1,44,000 ligands were docked. Then the 14 top ligands we have identified, and then these 14 compounds were tested in the further. Now, to tell you just the thing or how exactly this project is. So it was not me who did with this 1,44,000 ligands docking. So it was a previous collaborator collaborated with another person, and then we got these 14 compounds. Now the challenge that was given to me is like we have these 14 compounds and we cannot test all the 14 compounds for the animal testing. So we got to rank, or you need to rank the compounds and then say like top 3 or 4 compounds, because we have an option to test only 4, not all 14. So rank them, prioritize, and then tell me which ligand I should use for the wet lab aspect. Now, when you have like 14 and to come up with 14 to 4, and then that goes next into the animal testing, right? So it's a very challenging task to be worked with. So in that context, I have to understand with this FFAR2 protein and then where exactly the active site and all those things, then perform the docking, and we got the energies like this for it. This is the VINA score. So -6, -6.1, -5.7, and the different energy values that are there between these two. We also I got to understand how exactly the active site is. Again, if you can remember whatever we discussed with that NARL protein, right? So for this FFAR2, there are three key residues that are there. One is this arginine 180 and arginine 255 and the histidine 242. Now, if we also look into this table, we have the small chain fatty acids as butyrate and acetate, and this cat PB is the antagonist. So these two are the agonists. This one is the antagonist. So we did docking and then we checked where exactly this butyrate binds, where exactly acetate binds, how is the energy, where is this cat PB is going to bind, what is its energy, how is the interaction. Now, if you imagine these two are the agonists, this one is the antagonist. So the property of the agonist is to activate the protein. The property of the antagonist is to stop the activity. So that means they work completely different way. And if I say like something like this particular ligand is going to activate the protein, and if they take into the wet lab and when they test, if it is going to inhibit the protein, then it goes in a completely opposite manner. So again, one has to check what is the property of the agonist and what is the property of the antagonist. How do they interact? How is this ligand is going to interact? Then we can take a decision. So to understand that, I did multiple times the docking to understand where exactly this acetate butyrate is going to bind, how exactly the cat PB is going to bind, how many hydrogen bond formation is going to happen. But this is not from our side. This paper was published in 2011 by another group where they mentioned like this is an agonist which forms two hydrogen or multiple hydrogen bonds. But the key important aspect is it forms two hydrogen bond, one with arginine 180 and another one with arginine 255. And this histidine 242 is helps in the, that is this one. So this supports from the backside just to make the stability for the ligand to stay there. So after understanding all this, so I took all the ligands, this acetate, butyrate, cat PB, and all the 14 ligands that they have provided, then looked into the interaction, how exactly the interaction of each of these ligands is happening. Is it just with histidine or whether it's interacting with arginine 255 or arginine 180, and then made a table. So here, if you look into, we also made it like it becomes like an agonist if interacts with both arginine 180 and arginine 255. If it interacts either with 180 or arginine 255, it becomes like antagonist. Now this helps. Now this helps us in making a decision like whether it acts like agonist or antagonist. Now we made it like 20% based on this interaction. I just, it's a completely intuition. Okay, so this is the kind of interaction, how many nine conformations and how many times it repeated. So I made it like 20% can act like an agonist. It cannot act like an antagonist because it is not interacting with these residues. 30%, 10%, 70%, 50%, 30, 20, 10. There are a few ligands which are like 30% agonist and 70% antagonist. So this is the completely an intuition based on the interaction, how is the complex looks like. I just made this table. Then for mouse FFAR3 also, FFAR2 also the same thing, analysis of all the conformation and then making it like whether this can act like an antagonist or agonist. Right, so here 40% as an agonist, 60% as agonist, 50% antagonist. Both the properties is there with this ligand, a kind of it. Then they took that information. Okay, so here Okay, so there was some internet issue. I think should be okay. So we did with the 3D structure evaluation. Then I checked how exactly the arginine 255 or arginine 180, how is the interaction and all those things for all 14 ligands. And we checked how is the interaction, basically where it binds, wherever the acetate or butyrate is binding, whether it's there in the same site or it has gone to different site, all those critical analysis. Then we made a decision of that this particular table, whatever I have mentioned. The 2D diagrams are also generated for each ligand, the 9 conformational binding site interactions, then check how is the interaction. Then the decision was made like that 40% or 50% agonist and antagonist. So based on that particular table, okay, so our collaborator, so he is the Hariom Yadav is the collaborator. He tested uh, with the binding of this free fatty acid receptor 2, FFR2, and then found out that the Fenchol, the compound which is there, commonly present in basil plant, is having a potential FFR2 stimulator. So even when we cross-checked with the table, whatever we have, right? So this is the Fenchol, right? So what I in the table, whatever I made is it is going to act 50% as an agonist, right? And it is not going to have any antagonist activity. So, but there are few other ligands. If you look into, right, this is another ligand which I made it like 70% agonist, but did not work out. So there is another ligand, right? This is 30% agonist, but 60% antagonist. Not to consider, not to consider, not to consider. This is also not to consider because 10 and 80. So when they tested with this and right few ligands where so Fenchol has come out to be a good promising lead molecule. So here with the mouse also, because with the mouse here I mentioned like 40% as an agonist, right, and 0% as an antagonist. So when they tested there also it proved to be a uh, good activator. So then we could make out a publication that's the activation of microbiota sensing if we are to signaling which ameliorates amyloid beta induced. And this we could publish in a good journal like Frontiers in Aging Neuroscience, having around 5 impact factor. Then before going for this publication, we made a review paper about FFAR2 and FFAR3 proteins. This also we could publish in MDPI Biomedicine Journal. Then our work could also be made into a presentation in Fenchol ameliorates Alzheimer's disease in the Alzheimer's and dementia uh, experimental molecular biology conference. And then this abstract got published in Journal of the Alzheimer's and dementia, having like 16 impact factor. Then this work got uh, in all the newspapers and magazines there also in the US. So Alzheimer's disease can be prevented. So it provided a hint. So overall, if we just look into right, so one is with a dry lab, and also if we have a good collaborator to test whatever you have done, right? So we can aim for publishing in high impact factor journals and can also uh, gives a clear hint like this is how it's going to work and helps in research. So thank you very much. If you have any doubts, I'll be more than happy to answer. Please unmute yourself, or you can also uh, send in the WhatsApp group. So if you have any questions, any participants with any of the steps or anything related to chemoinformatics, molecular docking or dynamic simulation related, if it is there, we'll be more than happy to answer. Okay, so if there are no questions, thank you. I think we'll just check with Dr. Dev, Dr. Dev, and our next session has not yet joined, so we can have our next session. I think I will just check his availability. If he's available, we can have it from 4 o'clock to 5:30 from the. Thank you.